In our previous lesson, we learned about the four-step process of aerobic cellular respiration. So we began in the cytoplasm of our cell with the first step, which was glycolysis, followed by the formation of acetyl coenzyme A, 2 turns of the Krebs cycle, both of these steps taking place in the matrix of the mitochondrion, and then lastly, the final step taking place on the cristae, on the folds of the inner membrane of the mitochondrion, the process of chemiosmosis, oxidative phosphorylation, producing lots of ATP. And so after glycolysis, 2 turns of the Krebs cycle, and a lot of ATP production in the final step, we ended up with a maximum output of 38 ATP. Well, all of this was predicated on the assumption that we were starting with glucose, that the molecule we were using at the beginning of this process was glucose. And glucose certainly is the primary fuel that our cells burn through aerobic respiration whenever we need to make ATP. But surely glucose is not the only fuel we can burn. I mean, how often do you sit down at the dinner table and say, "Mom, would you please pass the glucose?" I mean, you don't eat glucose, at least not directly. So there must be other processes that can convert some of the things that we eat into glucose. And certainly there must be other fuels besides glucose that can be burned in this aerobic pathway. That's what we're going to discuss today. I'm sort of calling this the versatility of catabolism, or I could have called it the versatility of aerobic cellular respiration. This is still part of our unit on cellular energetics. Specifically today we're covering concept 7.6 in the Campbell Biology in Focus textbook. That's pages 157 and 158. So this is the diagram that we looked at last time of the 4-step aerobic respiration pathway. Here's step 1, glycolysis. Notice I started with glucose, and we talked about how in the first half of glycolysis, glucose is split in half to make 2 3-carbon G3P molecules, which are then converted into pyruvate. And then we talked about those 2 pyruvates moving into the mitochondrion and getting converted into acetate with the loss of carbon. And then we followed those 2 acetates as they moved through the Krebs cycle and so forth. But again, we were assuming that we're starting with glucose, that glucose is the fuel that our cells are going to always be using whenever they do aerobic respiration. And that's simply not always the case. It turns out there are other starting points that your cells can use to make acetate. There are other molecules that can be used to make pyruvate. You don't always have to start with glucose. Glucose is the primary fuel that our cells burn. And glucose is often readily available because, as you know, glucose is in our blood. We refer to that as blood sugar or blood glucose. And the reason it's present in our bloodstream is because it is the primary, the main fuel that our cells burn. But it's not the only fuel. Okay, so we're going to investigate some of these other fuels today. So glucose is not the only food source that can be oxidized for energy in aerobic cellular respiration. Other sources of energy are carbohydrates. Now, of course, glucose is a carbohydrate, but we're going to talk about how other carbohydrates like starch or glycogen can be used as the starting points for respiration. We're going to look at how proteins can be used as fuels and also fats. So those three, with carbohydrates being the main source of energy, but carbs, proteins, and fats all can be used as fuel, and they all can be broken down in some manner by the aerobic respiratory pathway in order to make ATP. So let's start with the easiest thing, and that would be carbohydrates. I say easiest just because every carbohydrate can either directly or indirectly be converted into glucose. So, for example, when I eat bread or potatoes or pasta, all of those foods contain starch. And if you remember anything from Unit 1, you remember that starch is a polymer of glucose. A molecule of starch is nothing more than a glucose bonded to a glucose, bonded to a glucose, bonded to a glucose. It's a long chain of glucose. So it's very easy when I eat starch, it's very easy for my body, the amylase enzyme in my saliva and also in my intestines, that enzyme can very easily chop up that starch molecule and produce large amounts of glucose. We also have glycogen. Animals always have a supply of glycogen stored in the liver and in our muscles, and that's a way of storing glucose. Glycogen is also a polymer of glucose. So whenever we need some additional glucose, whenever we need to do aerobic cellular respiration, we can start by going to that glycogen that we have packed away, and we can break it down. Hydrolysis. We add water to break the bonds, and when you break up starch or glycogen, you get a whole bunch of glucose. You might say, well, what if I'm eating a food that doesn't have starch, but it has lactose? Maybe I'm drinking milk and so it has lactose, or I'm eating a sweet, a dessert item that has sucrose. Well, those are disaccharides, and most disaccharides contain glucose, or the disaccharides can be digested down to their monosaccharide form. In other words, the double sugars can be split in half to make single sugars. And then all monosaccharides can very easily be converted into glucose. So even if you're eating a food that contains a different monosaccharide, say something that has a lot of fructose, like an apple or a banana, those are going to be naturally sweet because they have the sugar fructose in them. Well, fructose can very easily be converted into glucose. So the bottom line, I'm taking a long time to make a very simple point. Any carbohydrate that you consume, once you digest it and break it down, it's very easy to convert that into the monosaccharide form and then into glucose specifically. So if we eat carbohydrates, which a lot of the foods that we eat contain carbohydrates, we can break those down into simpler sugars, and then those sugars can be converted into glucose, and then we're just right back at, we just do normal aerobic cellular respiration where we run glycolysis, and then we convert pyruvate into acetyl-CoA, and then those two acetates go through the Krebs cycle just like normal, and we make the 38 ATPs. Okay, so no, you don't sit down at the dinner table and eat glucose. Probably never have you done that. But you eat carbohydrates all the time. Pasta, bread, and potatoes. And you simply digest those carbohydrates and turn them into glucose. So that is very often how we end up with the glucose that we burn in aerobic cellular respiration. Now let's suppose that I'm eating a nice juicy grilled steak, one of my favorite foods. Well, that steak is going to have a large amount of protein and also probably some fat content, but no carbohydrate. Now, if I have a baked potato with my steak, sure, it'll have carbohydrate. But what about the protein in the meat? Can I take any part of that protein and run it through the 4 steps of aerobic cellular respiration? Well, yes, I can. Now, there's no way to convert protein into glucose. You'll notice that none of the purple arrows point to glucose because there's no part of a protein that can be changed into glucose. However, it is possible to take a protein, break it down. What do you get when you break it down? You get a whole bunch of amino acids, right? Because that's what proteins are made of. And it is possible to convert amino acids, some of them, into pyruvate or acetate. And then you can run those acetate molecules through the Krebs cycle, because that's what the Krebs cycle does, right? It combines acetate with oxaloacetate to make citrate. Remember, that was the first step of the Krebs cycle. So the Krebs cycle doesn't care where the acetates come from. Normally they come from glucose following this pathway. But it's possible to take amino acids from a protein and turn those into acetate. And they can be plugged into the Krebs cycle. Again, the Krebs cycle is not picky. It doesn't care where the acetate comes from. So here's kind of what we're saying here. When I eat protein, a steak, and it gets into my stomach and my small intestine, the enzymes in my digestive tract will break down the protein, chop it up into its individual building blocks. What are the building blocks of proteins? Amino acids. What is an amino acid? Well, you're looking at one right here. And all amino acids have this little nitrogen part called the amino group, and they all have this part over here called the carboxyl group. And they all have a carbon in the middle called the central carbon, and a hydrogen here. And then there's 20 different things that can plug in this spot. Remember, this is called the R group right here, this part that I'm pointing to. But other than the R group, the rest of the amino acid, they always look the same. They look exactly like you see right here. Now, what do we do with those amino acids? Well, most of the time, nearly all of those amino acids are used not as a fuel, not to get energy, but they're used as building blocks to make new proteins. Okay? So think about it this way. When I eat a steak, I'm eating protein from cow muscle. Now, I'm not a cow, so I have no direct use for cow protein. My human cells cannot use cow protein. So what I must do is digest that cow protein, break it down into its amino acid components, and then my cells can take those amino acids and build my own human proteins, my muscle proteins, my enzymes, whatever kind of proteins I need to build. I can use the same building blocks that were in that cow protein, and now I can use them to build my own protein. And that's what you're going to do with most amino acids. However, sometimes if you have exhausted your supply of glucose and your cell does not have access to any glycogen that can be broken down to make more glucose, sometimes proteins, or I should say amino acids, these things, can be used as an alternative fuel. Usually, when you run out of carbohydrate, you would go to fats. Fats would kind of be your secondary fuel, and protein is kind of your last ditch effort. It's kind of the last possible choice for fuel. But amino acids from proteins can be used as fuel for aerobic cellular respiration. However, whenever you're going to use an amino acid as fuel, this part, which is called the amino group, has to be removed. You can't really do anything with the nitrogen part. You're only really interested in the carbon part of the amino acid. That's where the energy is going to come from. So the nitrogen part has to be removed. That process is called deamination. And these leftover nitrogens, these leftover amino groups, become what we call nitrogenous waste. It kind of builds up and becomes a toxin inside the cell. And so our kidneys filter out that excess nitrogen and convert it into either ammonia or urea, and that's where PP comes from. Okay, that's the main ingredient in urine. The reason urine has urea and small amounts of ammonia is because of this process of removing the nitrogen part of an amino acid so that the remaining carbon part can be used as fuel. Now, some amino acids only have 2 carbons. Sometimes the R group doesn't have a carbon right here in this spot. So if it's an amino acid that only has 2 carbons, those amino acids are converted into acetate. Acetate is a 2-carbon molecule, and we can plug that acetate directly into the Krebs cycle. However, if it's an amino acid that does have at least 3 carbons, like this one does, then we can convert that into pyruvate. And pyruvate is a 3-carbon compound. Remember, normally we make pyruvate through glycolysis, through splitting glucose in half. But this is just another way to make a 3-carbon pyruvate by simply modifying these 3 carbons from an amino acid. And you would simply take that pyruvate now, remove one of its carbons, and turn it into acetate. Okay, so let's look at it. I know this is a lot to take in. It's a little confusing. So I'm going to digest my protein. I'm going to break it down into amino acids. If it's a 2-carbon amino acid, I'm going to turn it into acetate, and I'll simply run it through the Krebs cycle. If it's a 3-carbon amino acid, I'll turn it into pyruvate, and I'll begin the process with the formation of acetyl-CoA. I can't start here because I'm not starting with glucose, and there's no way to turn a protein into glucose. So what I'm going to do then is just bypass glycolysis, and I'm going to take that amino acid and either turn it into a pyruvate or turn it into an acetate, plug it into the Krebs cycle. So as I said before, your enzymes in your cells, in your mitochondria, they're not picky. They don't care where pyruvate comes from. Normally we get pyruvate by splitting glucose. You split glucose in half, you get 2 molecules of G3P, and then you just convert those into pyruvate. That's the normal way to make pyruvate. But another way to make it is to take a 3-carbon amino acid and convert it into pyruvate. So it doesn't matter where the pyruvate comes from. I can still take any pyruvate, remove the carboxyl group, turn it into a 2-carbon acetate, and then plug it into the Krebs cycle. And every time I run the Krebs cycle, every time an acetate enters the Krebs cycle, I'm going to get 3 NADHs, 1 FADH2, and 1 ATP. It doesn't matter where that acetate comes from. If it comes from glucose, that's fine. If it comes from glucose, I'm going to have 2 acetates, and I can run the Krebs cycle twice. Okay? But if I only have 1 acetate, that I made from a 2-carbon amino acid, so what? I'll just run the Krebs cycle one time. I'm not going to get as many ATPs, but I'm still going to get some. So the whole point of this is that this process is versatile. It normally starts with glucose, but it doesn't have to. It doesn't have to start with glucose. It can start with pyruvate made from an amino acid. It can start with acetate made from an amino acid. But what if the fuel that I want to burn is a fat? Well, fats can be used as fuels. In fact, stored body fat is the most highly concentrated source of stored energy in an animal's body. There's a lot more potential ATP that can be made from a gram of body fat compared to, say, a gram of glucose. There's no contest. Fat is a very energy rich compound. But obviously, when you break down fat, when you digest fat, either breaking down your own stored body fat or digesting the fat that you eat in a cheeseburger, there's no way to convert that fat into glucose because fat is not a carbohydrate. So what do you get when you break down fat? Well, fat is a triglyceride. So I should have put a picture of a triglyceride on here. I didn't even think about it. Actually, I'm going to pause it and I'm going to put a picture on here. Okay, so I'm back now. Look, magic. There it is. There's a triglyceride. So this is one molecule of fat. All right. So when you break it down, when you digest that, what do you end up with? Well, this part here, this 3-carbon backbone, is called glycerol. It's actually a 3-carbon alcohol. And then these long chains of carbon are called fatty acids. So this would be a saturated fatty acid. Here's a saturated fatty acid. You remember what you call it when it has a double bond in between the 2 carbons and it kind of has a little kink? Remember, this would be an unsaturated fatty acid. Well, none of this is going to turn into glucose. But I could take these 3 carbons right here from this glycerol and I could turn them into a 3-carbon compound called G3P. The very same G3P that's normally made in the first half of glycolysis. The very same kind of G3P that normally is made when I split glucose in half. This is just another way to make it. I could take these 3 carbons right here and convert them into a molecule of G3P. And then I could run that through the second half of glycolysis and then run it through the rest of, formation of acetyl-CoA, Krebs cycle, electron transport chain. And then I've still got these 3 fatty acids, these long chains of carbon, 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 carbon. Fatty acids are sometimes called hydrocarbon chains. I can't turn them into glucose, but what I can do is chop them up. You see how I could take these 2 carbons and then break this bond, 2 more carbons, break a bond, 2 more carbons. I could chop this thing up into little 2-carbon chunks, 2-carbon fragments through a process called beta oxidation. And so each little 2-carbon chunk can now be converted into an acetate. So if you just try to sort of picture this, if I took these 2 carbons and made one acetate, then another acetate, 3, 4, 5, 6, 7, 8, 9, 10, I could probably make at least 15. I could probably make at least 15 molecules of acetate just by chopping up these 3 fatty acids. And some fatty acids are much, much, much longer. They just keep going and going and going. So you can actually make, at least in theory, up to 42 acetate molecules from a single molecule of fat. Now think about it. Every time you break down a glucose, how many acetates do you get? Two. Just two. And that means you can only run the Krebs cycle twice. But if I have a really large fat molecule with really long fatty acids, and I can get 42 acetates, that means I can run the Krebs cycle 42 times. Think about how many NADHs and FADHs and ultimately how many ATPs. This is just a rough estimate, but somewhere in the ballpark of 500 ATPs after running all of those Krebs cycles. The point is, fats can absolutely be used as fuels for aerobic cellular respiration. But there's going to be a lot of preliminary alterations that are going to have to be made. The reason fat takes so long to burn, if you've ever tried to lose weight, body fat can be notoriously stubborn, and it takes a lot of exercise for our bodies to break down fat. The reason is, just to process this body fat, I've got to separate the glycerol from the fatty acids. I've got to cut up all the fatty acids into 2 carbon chunks. Then I've got to chemically modify those chunks and turn them into acetates, and then plug them into the Krebs cycle. And so there's a lot of steps. It's a lot easier just to take glucose, split it in half, and and it's just so much faster. Glucose is a much more readily available fuel. Okay, so just to recap, if I'm burning fat, first I'm going to digest it and break it down into its 2 chemical components. Glycerol is the 3 carbon part of the fat, and that can be converted into G3P. This is PGAL is the old acronym, but most textbooks now call it G3P, glyceraldehyde 3-phosphate. And I just run the process from there. And then I'm going to take those 3 fatty acids and I'm going to chop them up into 2 carbon hunks. Okay, that process of chopping up the fatty acid is called beta oxidation. That's going to give me a whole bunch of little 2 carbon acetates, which I can plug in, boom, right there. Run them through the Krebs cycle. I can keep running that Krebs cycle until I run out of acetate molecules and make a whole lot of ATP. All right. So I think I've touched on everything. I didn't say a whole lot about this part of the diagram. I just wanted to go back to proteins for just a minute. I know we did talk about this, but I don't know that I pointed it out on the diagram. Whenever we use protein as a fuel and we digest the protein down into amino acids, remember there's a portion of that amino acid that we can't use. That's the nitrogen part. And so they're showing us here we're going to have to release that nitrogen part off of the amino acid. That's called deamination. That's going to generate a bunch of excess sort of refuse, like garbage nitrogen that our body has to get rid of. Most animals either convert that into ammonia. NH3 is actually ammonia. Or mammals tend to mostly convert it into a very similar compound called urea. And those are the 2 chemicals that get filtered out by our kidneys, and that's what makes urine. So I know I mentioned that, but I didn't really show it to you on the diagram, so I just wanted to go back and touch on that. Just to recap today, glucose is normally the starting point for aerobic cellular respiration. But these other sources, these other starting points can be utilized. With carbohydrates, it's easy. You just turn it into glucose. With proteins and fats, it's not quite as easy because you can't turn those into glucose. But you can turn amino acids into pyruvate or acetate. And you can turn fat into pyruvate or acetate. You just have to do a lot of preliminary processing first. So that concludes our lesson for today. As always, it went longer than I thought it would, but I hope you learned something, and I hope to see you real soon. Take care.